Hello everyone. I believe you all have heard the story of Dolly the cloned sheep. This story, once published, caused a global sensation and discussion. The birth of Dolly not only challenged the traditional concepts in the scientific community regarding cell differentiation and genetics, but also sparked a series of discussions on the ethical, legal, and social implications of cloning technology. So, how was Dolly cloned? How is she different from a normally born lamb? To answer these questions, we need to start with cells. We know that animal cells come in two types: somatic cells and reproductive cells. Somatic cells make up the tissues and organs of animals, such as epithelial cells of the skin, hepatocytes of the liver, etc. These are highly differentiated cells containing a complete set of genetic material, specifically 23 pairs of chromosomes, which means they contain a complete set of DNA genetic material. Reproductive cells, on the other hand, are divided into sperm and eggs. Sperm comes from male animals and eggs come from female animals, each containing half of the genetic material of a complete cell, which is 13 chromosomes. Normally, when a female animal's egg meets a male animal's sperm, they combine to form a fertilized egg. This fertilized egg contains a complete set of genetic material and can gradually develop into a new organism in the female animal's uterus, eventually giving birth to a new life. But does this mean that all cells containing complete genetic material can develop into a new organism? The answer is no. Most somatic cell nuclei in adult animals contain complete genetic material, but cannot develop into a new organism. So, can the nucleus of somatic cells of adult animals develop into a new organism under specific conditions? If this could achieve the birth of a new organism, wouldn't humans be able to constantly replicate themselves? To answer this question, we must first understand the biological cloning technology. Simply put, it involves the following three steps. 1. Extract the nucleus from the somatic cells of an adult animal and transplant it into an enucleated egg cell of a female animal. At this point, the nucleus of the modified egg cell has been replaced by the nucleus of the somatic cell of an adult animal. 2. Culture the modified egg cell and then implant it into a surrogate female animal. 3. After a normal gestation period, a new life is born. Dr. Ian Wilmut and his team used cloning technology to transfer the mammary gland cell nucleus of an adult Finn Dorset sheep into the enucleated egg cell of a Scottish blackface sheep. Then they implanted it into the surrogate mother sheep. After the normal gestation period, Dolly the cloned sheep was successfully born. I have briefly explained the process of Dolly's formation in a few sentences, and it may seem that cloning a sheep is not difficult. But in fact, it is not so. The success of cloning Dolly was achieved after the relentless efforts of many scientists. As early as 1952, scientists successfully conducted a cell nucleus transplantation experiment on frogs, which was the first use of early embryo cells to produce clones. Later, in 1958, John Gordon used the nuclei of adult frog cells for nuclear transplantation, proving that the nuclei of adult somatic cells could also be used for cloning. Additionally, in 1975, scientists successfully created mammalian rabbit embryos using nuclear transplantation technology. In reality, Dolly was not the first cloned mammal. This honor belongs to another sheep cloned from embryonic cells, born in Cambridge, UK, in 1984. Before Dolly, the Roslin Institute had already cloned sheep named Megan and Morag from lab-cultured embryonic cells. However, these experiments were all conducted on lower animals and embryonic cells. Dolly the sheep is significant because she was the first mammal cloned from adult somatic cells. Her birth proved that highly differentiated adult mammalian cells could be used to create new life, a concept previously deemed impossible. The researchers' ability to reprogram differentiated cells indicates that these cells do not undergo irreversible genetic changes during development. This discovery profoundly impacts our understanding of genetic development and cell differentiation. Dolly the cloned sheep spent a short life of 6 years at the Roslin Institute. During these 6 years, she lived a normal life like the other sheep at the Institute, with occasional media appearances. Dolly and a Welsh mountain ram named David had 6 lambs. Her first lamb, Bonnie, was born in April 1998, followed by twins Sally and Rosie the next year, and triplets Lucy, Darcy, and Cotton in the following year. This showed that cloned animals could reproduce naturally. Dolly's offspring inherited genetic information from both her and her mate, exhibiting normal growth and development without being affected by Dolly being a clone. The health and performance of these offspring further confirmed the potential of cloning technology and its application value in biomedicine and agriculture. Thank you for watching.